Hi friends, cordially I welcome you all once again. Today we will be discussing interesting topic of hematology, namely RH blood group system in human beings. This interesting topic explains how a person is positive or negative in blood group. So let's talk about first of all discovery. This blood group was discovered by two prominent scientists, Carl Landsteiner and Alexander Wiener. They performed number of experiments. Initially, they selected a monkey, namely rhesus monkey, or we can say macaque rhesus. They transfused the monkey's blood into one more organism, namely rabbit. Now, what they admire, after proper transfusion, they found rabbit's blood. It shows cross reaction, or we can say agglutinization, against the monkey's blood. It is because one of the solid reason. In rabbit's blood, specific type of antibodies are formed. These antibodies are known as Rh type of antibodies, or we can say Rh anti-sera. You have already know that antigen antibody reaction takes place during transfusion. They tested 100 types of macaque rhesus monkey and found same result. Now it happens because one of the main reason RBC cell of the monkey contains specific type of antigen on its surface. Suppose this is RBC cell and this is the antigen. The antigen present on the RBC cell of monkey is called Rh antigen, or we can say rhesus antigen. Hence we can here conclude that rhesus monkey has a Rh antigen, so it is a Rh positive monkey. Now in the second experiment, they tested different types of human beings. They performed same type of experiment. They transfused blood of human beings into the rabbit. Again, a rabbit forms what we call it as Rh antibodies and shows agglutinization. Now they conclude here, because it happens one of the solid reason the RBC cell of the person has a Rh antigen, the same antigen that was present in the monkey. The person which has Rh antigen like that of monkey is known as Rh positive person, while as reverse of that is Rh negative, the person which lacks Rh antigen. Hope you got the discovery. Now let us discuss core concept of that very topic. Here we are going to define what is basically Rh type of antigen. The Rh antigen present in the human beings, it has about 50 types. But clinically, 5 are more important or most immunogenic, which evokes our immune system during transfusion. That means which are taken into consideration. Namely, capital D, one more, capital C, small c, and capital E, small e. These 5 types of Rh antigens, they are known as Rh family, or we can say Rh factors. These 5 types of Rh antigens are controlled by number of genes. The genes involved here are Rh capital D, number 1, Rh capital C, and last one Rh capital E. So these genes are responsible for the expression of Rh antigen. Now these genes are present on chromosome number 1 in human beings. Here let me show you basic structure of Rh antigen. Suppose this is erythroblastic precursor cell, means underdeveloped RBC having well-defined nucleus, and it has 12 post membrane protein polypeptide chain because it passes 12 times through the membrane. And this polypeptide chain, it has, this is amine group towards inner side, and one more side it has carboxylic group in it. These two groups lie towards intracellular surfaces. Right? Now look towards extracellular surface of that very RBC cell membrane. These loops are extracellular loops, and it has antigen determining property. This loop contains all Rh type of antigens D, one more capital C or small c. On next loop there is capital E, or we can say small e. collectively these 5 antigens are known as Rh antigens, which indicates when we talk about Rh antigen, it is a combination of 5 antigens that already we have mentioned. Alright? Among these 5 antigens, D antigen is very important to understand. This D antigen is most immunogenic and takes into consideration while transfusion. It is most immunogenic in nature. In other words, we can say Rh antigen is determined by D antigen alone. Hence, we can conclude here, the person who is Rh positive is also called D positive. The inverse of it says the person which is Rh negative is also known as D negative. Now, if we go for detailed explanation of the D antigen, the complete D antigen has well-defined structure. It has 4 epitopes. Let's assume D means dancer. Suppose this is the diagram of good dancer. A complete dancer has 2 arms, number 1 and 2nd, and 2 legs. This is number 3rd and 4th one. Presence of all epitopes makes it good dancer or good antigen. Now let's contrast what is Rh positive and Rh negative type of blood group system. How we can say person is Rh positive or Rh negative. Now for good explanation, let us consider number of RBC of different persons. Suppose this is RBC cell of person number 1. It has all D antigens in good concentration present. So we can call this person simply as Rh positive type of blood group person. Now in next, what happened? Here all Rh antigens are absent, so it is simply Rh negative type of blood group. Now there are few more persons. Suppose this is RBC cell of 2 more persons. Here the person has RBC cell having D antigen but in low concentration. We can call this person simply it has a blood group but weak Rh type. Now talking to the next person, this person has RBC cell having D antigen but not complete D. It has broken legs, broken epitopes. The person is said to be having partial Rh type of blood group system. Hence we reached conclusion and we can say Rh positive and partial Rh, it shows qualitative difference. Accordingly, Rh positive and weak Rh, it shows what we called it as quantitative difference in the D antigen. Now there is one more type of Rh type of blood group system. Suppose this is RBC cell membrane. This person lacks Rh antigen, so we can say it is a Rh null or golden type of blood group system. It is very rare in nature. Basically, difference between Rh null, Rh negative is Rh null lacks all type of 5 antigens, while as Rh negative lacks only D antigen. Rest 4 antigens are present. Hope you got this. Now in this part we are going to contrast and compare A, B, O and Rh type of blood group system in the human beings. Now let me write here different types of antigens, different types of phenotypes, or we can say blood groups, and their genotypes present in the human body. Suppose there is a person which has antigen A present in the RBC cell membrane. Okay, if it has only A antigen, the person has blood group A negative. Hope you got it. Now if the person has A antigen along with Rh antigen, now we call this person as A positive blood group. Hope you got this point. Accordingly, the person having B blood group only, it is B negative. The person having B along with Rh antigen on the RBC cell membrane, we call this person as B positive. Okay. Now one more case is a person has A and B, both type of antigens. It has AB negative type of blood group. And if a person has AB along with Rh type of antigen, we call this person AB positive blood group. This AB positive blood group acts as universal acceptor. Now at last, suppose there is a person which has which lacks all type of antigens, but it has only Rh antigen. We call this person O positive, right? And reverse of this, the person which lacks all antigens in the RBC cell membrane, this is known as O negative. That's why O negative is known as universal donor type of blood group. It is very rare type. Now in this chart, let me add one more point, genotype of different blood groups. A blood group has genotype either homo or heterozygous. B blood group has same homo or heterozygous. It shows incomplete dominance sometimes. And AB blood group shows codominance. Accordingly, O blood group has this type of genotype. Now let us add one more column, that is antibodies present in different types of blood groups. A blood group has B, accordingly B has A, AB blood group has nil, and O blood group has all antibodies present in it. It was all about RH type of blood group system. Hope you got the concept. Have a nice day.